clips from the BBC's Life in the Freezer. And obviously now they've gone on and done Frozen Planet and Frozen Planet 2. And I loved learning about it, was absolutely fascinated, but thought, there is no way I'd ever go to Antarctica. I can't stand the cold. And this is having been brought up in the UK, where it's fairly cold compared to Australia. But would you believe it? As soon as I finished my PhD, Antarctica is exactly where I ended up. An opportunity came up for a postdoctoral position, and I thought, well, why not? Let's apply. I probably won't get it. Because after all, you know, I was 22 years old. Anybody can come to Australia pretty much any time, provided there's not a global pandemic going on. But not everybody gets an opportunity to go to Antarctica. So I ended up, 22 years old, tens of thousands of miles away from my home in the UK, in Antarctica. So you might ask, did I go wrong? Did I lose my way? And I'd say, maybe, but no. I took a risk. I took a chance. And it's got me to where I am today. So here's Antarctica. And I've got my notes here, because just in case I forget to say what I'm going to say, because we don't have presenter view. But I want to tell you a bit about my early career. But first of all, I'm going to get you to put your thinking caps on your imagination, because I want you to imagine a jumble of buildings in an area that's much smaller than the campus that you've been wandering around and working on, probably a quarter of the size of this campus. And they're all different colours, a bit like a Legoland. I want you, given you've spent 2 weeks together, you've got to know each other quite well. I want you to pick about 20 to 25 of the people you've got to know that you want to go and spend time in Antarctica with. So have a think about it, look around the room if you want. But there's a bit of a catch. They can't just be the people you like. At this point, don't look at people, please. But if there's someone that annoys you because they chew too loudly, or they talk too much, or they snore in their sleep, you've got to take 2 or 3 of them too. Is there anyone who wants to be a doctor, a medical doctor? Hands up. Okay, from these people who've got their hands up, pick one of them, because they're coming with you too, because you need a doctor to look after you, but you also need a doctor to sort of study your ever-decreasing grip on reality as you winter in Antarctica. And I'm not joking. And then is there one of the teachers or mentors that you've had this week who's really inspired you and been a great leader? Oh, that's very nice. I was going to say don't name names and single people out, but just keep them in your mind, because they have to come too, because you need a station leader who's going to keep everything in check when things get tough. So that's what ended up with me. I spent 16 months doing a winter uninterrupted. I didn't come home, at a station, an Australian station called Davis. And Davis Station is here on the map. There are various other places marked here, and I'll come back to this map. But Davis, the Australians have 3 different stations on the continent: Mawson, Davis, and Casey here. And I was based at Davis. And if it wasn't certifiable enough to do one winter for 16 months, I did go back again and do another one for 13 months uninterrupted later on in my career. So this is what the station actually looked like, a veritable Legoland of brightly coloured buildings. It's known as the Riviera of the South, because even though it's freezing cold, it's actually one of the sunnier, less windy places compared to, say, Mawson Station, which is very well known for its blizzards. It's one of the most southerly, well, it is the most southerly Australian station at 68 degrees south, and it's managed by the Australian Antarctic Division, where I now work. And there's often a summer maximum of sort of up to 120 people can be coming and going through the summer, but in winter we're down to maybe 20, 24 people. So my first winter, there were 19 of us, and only 2 of us identified as female. And the second winter, there were 24 of us, and only 3 identified as female. So it was a very different environment to what you have in the real world. And this is the wintering party. And you can spot me on the end, looking a great deal younger than I do now. So what was I doing in Antarctica? Well, Davis Station is based in the Vestfold Hills, which is the third largest ice-free area in Antarctica. Unlike you might think, the whole of Antarctica isn't just ice. Most of it is, but there are around the edge of the continent, there are some ice-free oases, which are absolutely teeming with life. The area around Davis Station is about 400 kilometers squared, and it's absolutely spectacular. You can see here hills, you can see these black doleritic dikes that cut the landscape, so the geology is absolutely incredible, and these beautiful lakes that intersect that. There are over 200 lakes in the area, the small area. These are the result of isostatic uplift after the last ice age. And what I mean by that is when the weight of the ice melted and retreated, the weight of the ice came off the land and it started to rise. And it also scoured pockets in the rock. And those basins either filled with meltwater and became very fresh lakes, or they trapped relic seawater, so they were salty lakes. So now what you end up with is a range of lakes. This is Crooked Lake. This is one of the purest lakes in the world, freshest water. And it has a permanent transparent ice core that's over 2 meters deep, so it never melts out. And it's absolutely stunning to work on. But then at the other end of the spectrum, you've got ultra salty lakes like Deep Lake. This is 7 times saltier than seawater, and it's never known to completely freeze, even though the water temperature might go down to minus 20 degrees centigrade in the winter. But even so, it's still packed with life. Now, this is where what I was doing in Antarctica comes in. I know it's not charismatic penguins and seals that you've all heard about, but I'm going to convince you that microbes and tiny, tiny things are equally as charismatic as those penguins and seals. So I spent my PhD and then subsequent time as a postdoctoral scientist studying microbial food webs in the lakes, but also in the surrounding marine area. And this is a very simplified diagram of a very simple food web in those lakes. So at the base of the food web, we've got bacteria and viruses, millions of them, hundreds of thousands of bacteria in little vials of water, millions of viruses in that same vial. You can see them here on this picture, maybe not so easily with the lights up, but the longer rods and bigger dots of the bacteria, and then these smaller dots of virus, well, we call them virus-like particles. And the only reason you can see them is because, one, I'm using a powerful microscope, because they're absolutely tiny. I mean, they're all of a couple of millionths of a meter long, but also because I've used what's called a nucleic acid stain, which binds to the DNA and RNA of the bacteria and viruses and makes them fluoresce when I put UV light down the microscope onto them so I can count them. And I spent thousands of hours counting these things for many years. And just to make the point, on the pinhead, you'd fit 1 million bacteria. So you can imagine viruses are 100 times smaller. You're talking about 100 million virus-like particles sitting on a single pinhead, just for reference. So at the top of the food web, 1 to 2 meter long predators. Now, I think you're all probably familiar with water flea-like animals because we get them in lakes and ponds here as well. But you might get a sort of a sample of water, tens of milliliters, and there might be a few swimming around, and they're just visible to the naked eye. And I've put on the slide their winter pets, because we'd have them in the science labs in jars and culture vessels, and we very much kept them in our samples, and rather than sort of fixing them and killing them to count them, we would keep some swimming around over winter to keep us company. And we'd find that lots of people from around the station would deliberately come to the science labs just to see these, because there was nothing else to see other than human beings alive in Antarctica during the winter. Now, I sort of qualify that a little with the fact that the only 2 or 3 women on station were also in the science lab, so they may have been coming to see us, but probably the pets. Now, in between those bacteria and viruses and those top predators, we've got protozoa, which are single-celled organisms, animals and plants, that are pretty funky too. This is the images I'm going to show you next. There's 3 slides, and they're from scanning electron microscopes, where you do some heavy metal staining of cells, and you blow them up hundreds of thousands of times so you can see them. In fact, this little cryptophyte called Geminigera cryophyllum is only about 20 microns, or 20 millionths of a meter long. But it's very, very cool, because in the summer, it behaves like a little plant. It's swimming around in the water in the lakes. The lakes that I was studying, that ice cover does actually melt out in the summer, so there's a period where they get quite intense 24-hour daylight, and they can capitalize that and they can photosynthesize. And that's a process called autotrophy. I don't know if any of you have heard the terms autotrophy, where you can make your own food from sunlight, and then as opposed to heterotrophy, which is us, where you take something in to make it into your body mass. I'm seeing some nods around the room. Now, organisms like Geminigera, excuse my terrible cartoons, but in the summer, they're autotrophic and they photosynthesize. But in the winter, they're pretty cool because it's dark. There's 2 meters of ice on top of them. It's dark 24 hours. There's nothing for them to eat unless they find another strategy. And so they do. And one of the strategies is to take up dissolved organic carbon from the water around them and switch to a heterotrophic way of eating. Now, that wouldn't happen in your pond in your garden. It only happens because of the extreme conditions in Antarctica. So there's a rather silly cartoon of them sucking up with a straw, but the equivalent for us would be us sunbathing all summer and getting all our energy from the sun, and then in the winter having a really high protein shake to keep us going, okay? And it's a term called mixotrophy, where you mix the trophic strategies or the way you eat. So that's a term, if anything you take away from this lecture, that term mixotrophy is an important one. This is another of those little protozoans. This one's got 4 flagellae or tails that you can see. And this was the focus of my studies. And it's even more fascinating because in the summer it's a plant, it's using the sunlight. But in the winter, it doesn't just slurp up carbon. It actually becomes like Pac-Man, and that shows my age, doesn't it? Pac-Man going around eating bacteria. So some of these algae become carnivorous plants, and that's the way they survive. But they can also hedge their bets. So you'll find a portion of the population will, what we call encyst. They coat themselves in a hard shell and sink to the bottom of the lake into the sediments, and that's their survival. Sort of they're hedging their bets that if something goes wrong in the water column, there's still some of them alive sitting there to emerge in the summer. But a lot of them keep grazing all winter. So you'd find a very active community of algae, even in the pitch black under the ice. And then even better than that, there's another creature. This is a ciliated protozoan. So again, it's single-celled, and it's called ciliated because it's got all these cilias around it. And it looks for all the world like a hula skirt in that electron microscope photo. And they're even really beautiful under a light microscope, which is here when you stain them with something called Lugol's iodine, which stains the starch. It always makes me think about little sunflowers sitting, and you can easily count them under the microscope. Now, mesodinium's found all around the world, not just Antarctica. And if you've ever heard of red tides, where you get massive blooms of algae that stain the ocean red, and can cause all sorts of problems for fish and other species. Mesodynium is responsible for that. Now, Mesodynium does very well because not only is it mixotrophic and, not only is it heterotrophic and goes and eats bacteria, it can be considered mixotrophic because what it does is it steals the chloroplasts from the algae that it eats. So it digests the rest of the algae, keeps the chloroplasts intact. And then in the summer months, it can use those chloroplasts to harness the sunlight as well and benefit from those. So they're incredibly clever little plants and animals, and far more diverse in the way they eat than, say, a penguin or a seal. So if we come back to that food web, based on what I've told you now, I can add some arrows to it. I can add this red arrow, which is the bacteria feeding the algae in the winter. And I can add a green arrow, which is ciliated protozoa like Mesodynium, using sunlight as well as grazing to survive as well. Now, one of the things to note, which is very interesting, is that throughout my PhD and beyond, I did try and culture some of these algae and force them to be mixotrophic in the lab back here in Australia, but found it completely impossible. It must be some kind of combination of that prolonged dark and cold and the really deprived conditions there are that makes them switch. If they're at all comfortable in a laboratory, they just don't want to switch their nutritional strategies. So that brings me to another tip, that skills are transferable. Because obviously, I learned quite a lot about microbial ecology when I was doing my PhD. But what I also learned was field work, how to survive in the field, how to work with a balaclava and thick gloves and all this clothing that I could barely move and wade through snow. How to work in a lab in Antarctica and many other places as well. I learned to work as part of a team, which is absolutely key. You can't do any of this work on your own. You can't go to Antarctica with a massive team supporting you and the science that you're trying to do. And I also learned how to handle data, crunch data, write papers, all skills that are transferable. And anybody who lives in Antarctica will tell you about the A factor. Not only are you contending with all that clothing and the fact that in this photo, this is me, and you go outside and your hair's instantly frozen to your face and to your balaclava. So you can imagine if I rip off that balaclava now, I'm going to lose half my hair. And if you zoom in, you can see my eyelashes are frozen too, and I can guarantee I've got snotsicles because my nose is frozen inside as well, because we're working at up to -40. And it's pretty hard going. But there's also the fact that you're contending with really hard conditions. So if something's going to go wrong, it's going to go wrong, and it always goes wrong when there's a blizzard going on, when you're trying to wade around the station or out in the field and you can't see what you're doing. And sometimes things are completely catastrophic. You could be making your pancakes in a tent. This is a polar pyramid or a Scott tent, very like the ones that Scott would have taken on his expeditions. They're great for living and working in. You can stand up in them, you can cook in them. But the next minute, your tent might be on fire. Now, I survived this because obviously I'm giving you a lecture, but I learned a lot the hard way through that experience. And I'm not going to tell you about how it happened and what went on now, but we can do that over dinner and questions, and I'm happy to take questions. But you can see I learned a lot more than just science, and that's a lesson for you. Remember to look up, find out what else you're learning, even when your experiment's going wrong. You are learning something by things going wrong, okay? And you're learning something by taking risks, even if they get you in a situation you don't actually want to be in. And those transferable skills and your passion for what you're doing could take you anywhere. Some of my colleagues who are the most successful are ones who've taken, for example, a molecular technique or stable isotope techniques or a statistical brain, and they've let it apply that to anything. So I've had friends who've been great with working with stable isotopes. And you'd think they'd be stuck in a lab somewhere, but they've taken it to Antarctica, they've taken it to study hippos in Africa, they've taken it to Scotland to study fish. They've traveled all around the world with that. And so it really can take you somewhere. And that's what happened to me. I'd had enough after my PhD in Antarctica. I'd really had enough for spending thousands of hours away from home staring down a microscope. Much as I loved Antarctica, I wanted to go and test my legs back in the tropics where I'd originally thought I'd go. So I ended up doing my first postdoc based in Canada, but with a conservation organization called Project Seahorse. So we're going to switch tack completely now, and I'm going to tell you a little bit about seahorses because they're super cool too. So seahorses, you may already know this, but they've got a bony skeleton, which is relevant to some of the stuff I'm going to talk about later. They've got a horse-like a head, hence the name, the genus Hippocampus, because hippo is horse. So they've got a horse-like head. They've got a mouth like a straw. They suck up their food. They've got eyes like a chameleon, so they can look around and ambush their prey while they're hiding in the seagrass or the coral. They've got a pouch like a kangaroo, which will become apparent why they have that now. And they've got a tail like a monkey because they hang on to things. They're not very good swimmers, so they have to hang on to a holdfast like coral or seagrass or whatever else they can hang on to. And so, you know, just in case you're in any doubt, they're a fish. But there's lots of tales of sea monsters and in history where people had no idea what they were. They vary in size. For those of you who live in Australia and you might have been snorkeling, you might have seen them. The biggest seahorse is the pot-bellied seahorse, Hippocampus abdominalis, and it's up to 1 foot long, so that's one of your rulers for school. And they're absolutely incredible creatures to see swimming around. But then the smallest one is these tiny, tiny pygmy seahorses. This is Hippocampus bargibanti, and they hide in deep sea or deepish sea Gorgonian corals, and you can barely spot them. They're so well camouflaged. Tiny, tiny little things. And you measure the height of a seahorse, just an anecdote here, from that coronet, the top of the head, down to the tip of the tail. Now, seahorses, does anyone know why seahorses are pretty famous? What's special about them? I should be looking at the males in the room. Go ahead. The males give birth. So the males not only give birth, but they're the ones who are pregnant. They brood the young, carry them around for weeks to months, and then they give birth. So we're going to see, this is where the technology will probably fail me. Let's see if this link works. Ooh, well, it didn't, did it? It's supposed to be embedded. No. I will try and leave it. We did set it up and embed it to see if it would work, but I'm just going to go out and then, let's see, hyperlink. It was linked. Gabe managed to get it to work, but let me see, because it's just a cool video, but I don't want to waste too much time on it. Now I've got to find our good friend Google. Help, anybody? Where do you want me to go? Top left, over there. Oh, yeah. And then put it in. Hello. There we are. Let's see if I can get it to work. Oh, no. I'm hopeless. I'm getting performance anxiety here, because you're going to watch me flail around with the technology. Paste. Let's see. Did it go? Did it go? Yes. Right. I'm sorry. So male seahorses can give birth to something between a couple of hundred and a couple of thousand baby seahorses. And they're all little tiny mirror images of their parents. And the male might have worked really hard to brood them, but he pretty much leaves them on their own as soon as they're born. A couple of thousand baby seahorses just get left in the ocean drifting around. And obviously, a lot of them don't survive because they're great food for other fish. Now, so that was all some interesting facts about seahorses, but I did actually do some research as well, and that, I think, I was speaking to someone earlier about whether I did diving or not, scuba diving or not, and yes, I did. I went to the Neisder Estuary in South Africa and spent, I think, about 6 weeks on a project with a small team, and we were looking for the most endangered species of seahorses in the world, not the Neisder seahorse or Hippocampus capensis for the Cape of South Africa. Now, we spent weeks underwater in very murky estuaries. It wasn't exotic, it wasn't tropical, there weren't coral reefs, it looked like this. Murky brown water and murky brown seahorses, and we had to spot them. And we were doing underwater scuba surveys, and we were counting them and working out, basically doing one of the first field surveys to find out how many of these seahorses were left. And we were the first sort of team to find out roughly how many there were. If you're interested, we estimated there were only about 89,000 of these fish in this estuary. And that sounds like quite a lot when you think, oh, well, there must be loads of estuaries with 89,000 in. There aren't. There are only 4 estuaries in South Africa, and this is the main one where those seahorses live. They're all under threat, extreme threat of development and human influence. It would not take much to wipe those seahorses out if there was a big oil spill or a big development happened, or a big event like a freshwater flood, which regularly happens if there's heavy rains, and then you get big fish kills. So they're heavily endangered and they need protection. And so this data helped to influence some of the protection measures that the South African government put in place. But apart from that science, what's really great about seahorses, and one of the reasons I went to Project Seahorse in the first place, is that seahorses, like some other iconic species you could think about, are great mascots for conservation, for marine conservation. There's some environments here. There are 47 species known of seahorses. They all live in shallow coastal habitats which are close to where humans live as well. And those habitats, like the coral reefs, the mangroves, the seagrass beds, and those estuaries, are potentially heavily impacted and under threat because of our activities. So by protecting the seahorse, which, you'd ought to admit they're pretty cute, charismatic animals, and they grab people's attention. If you're going to talk about a fish, a seahorse is a pretty cool one. Sharks are the same, they tend to grab people's attention. Then you can use that to protect the habitats. Likewise, if you care about coral reefs and you want to talk about protecting that, you naturally then protect the species that live there, which would include seahorses and others. So if you think about using the species you study to do some good, you can often use it to influence a lot more than you think. Now, one of the other great things about Project Seahorse is that we didn't just work on science. We did science research to inform it, but we also did active management of the trade in seahorses. So seahorses are, that bony skeleton I was telling you about, means they dry really easily. Millions, up to 20 million, are collected from the wild every year and dried to use for traditional medicines and souvenirs and curios like this. And that has a huge impact on wild populations. There's also a live trade, which used to come from the wild as well, but mainly from small subsistence fishery communities that would collect live seahorses to sell for a bit of extra money to feed families. Now we've got better culturing seahorses in captivity to feed the live trade, but still they're incredibly tricky animals to keep. That bony skeleton means they've got a very thin layer of skin which gets infected easily. They only eat and survive and thrive on live prey, good quality live prey. And so they're hard to keep. But of course, because they're really charismatic, you have them in your aquarium, you fall in love with them. If they die, you want to replace them, and you perpetuate a trade in these wild animals that probably shouldn't necessarily be in an aquarium. And another threat is bycatch. Can you spot the seahorse in that photo? Yeah? In there? Things like shrimp trawling, or I'd call them shrimp, you call them prawns here in Australia, is massively destructive. I'm sure you've heard about this before, that it's terrible for almost clear-cutting benthic environments in the ocean. But it means all those environments where these seahorses hang on to their holdfasts are, the seagrasses, the corals, rocky rubble, they're going to get caught. And if you look at this photo, quite apart from the seahorses, you can see that bycatch is an issue for so many other species as well. I mean, in there, okay, it's a prawn fishery, but what percentage of that catch do you think is prawns or shrimp? Can you see lots and lots of other animals that aren't shrimp, can't you, as well? So bycatch is a massive issue for seahorses and other species. But I'm coming to another tip now, that sometimes you have to walk away. It's great to take risks, but they might not take you where you want to be. They might not work out for you. It's okay to, as long as you spend a bit of time exploring it and trying it and trying to make it work, it's okay to go, no, that wasn't the right thing for me, and walk away. And I did walk away from working with seahorses, much as I loved it and could see a difference. For personal reasons, I had to leave Canada and come back to Europe and find another alternative path. So where did I walk to? I walked to Germany. I'm based in the UK, my parents are in the UK, but I went to Germany. And I went to an absolutely stunning place called Potsdam, which is not far from Berlin. And if anyone's been to Germany, there's Berlin, there's Potsdam where I lived. I worked at a university there. But the red star shows you the focal area of the studies that I was doing there, because this is where the extremely part of my talk title comes in. I've worked in the extreme Antarctic, but then I came back to work in Germany, and there's a region there called the Lausitz region, where there are a lot of lakes. They're lakes that have come from the mining. Has anybody heard of lignite, which is brown coal? Yeah, you've heard about it. So brown coal is very poor quality coal in terms of heat. It's only about 25% carbon of some of the other coals, so it doesn't burn hot. But it is used extensively for sort of steam energy production, and it was mined extensively in Germany. And that left lots of open cast mines, big pits in the environment that have gradually filled with water because of the rainwater and leaching. And some of the lakes, there are about 400 of these pits that are filled with water. About 26 of them, the last look I had, have been rehabilitated, and they're fresh and they're used for recreation, and they're actually, there are resorts built on the side of them, beautiful lakes you can go on holiday on. But the majority of them are still looking like this. And they're extremely acidic. They're extremely acidic because the lignite is very high in iron, and they've also got heavy metals in them and lots of calcium and magnesium. Cadmium's another one, and aluminium. So they're pretty toxic places. And they are this bright red color. These are my photos. Okay, they're not great quality because I'm not a great photographer, but they're this really red color because of the oxidization of the iron 2 into iron 3. And that's a theme that you'll see in some other forward slides. Now, working on these, your skin gets stained. My field clothes got stained. So do you think they'd be a good environment for things to live in? If you think of a neutral lake here in Australia or anywhere in the world, you're talking about anything between pH 6 to 8. And they're roughly neutral. These are down at pH 2.3, 2.6, incredibly acidic. So anything live in them? No?
Life always finds a way.
Well, obviously there's lots of dead tree stumps in there, so the trees don't live in them. But yes, there is a pelagic food web, and it's very, not similar in terms of the species, but it's very simple in the sense that it's very like those Antarctic food webs where you've got the bacteria and viruses down at the bottom, you've got some algae, these flagellates here, who are mixotrophic again, because that's the only way they can survive in the harsh conditions. You've got some ciliates that look a bit different from Mesodinium, but they're still ciliates. And then you've got some top predators, rotifers that go around eating things, and these heliozoa, which also go around trapping bacteria to eat and algae to eat. So things do live in them. Now, I told you this was a bit of a wild ride because I went from Germany then. While I was still living in Germany, I was invited to go to an Arctic research station because of my now sort of specialization in extreme environments. I went up to an island called Axel Heiberg Island, which is marked up there at the top. It's a rather large island up in the Canadian Arctic at 80 degrees north. And just for reference, you can see it's next to, there's Greenland there, and this is Baffin Island here, which you might have heard of. And I stayed at the McGill Antarctic Research Station, which is McGill University in Canada. And I was very much working there because this is where the astronaut bit, well, not astronaut, but space theme comes in. Because these field sites in the Arctic are some of the best analogues for space research on the planet. So if you're looking for life on other planets, you know, like Mars, where it's a very cold, harsh environment, and a desert in many ways, going to polar deserts, like the Arctic and some places in the Antarctic, is a great way to study and work out what you might find and what signs you might be looking for life. If you do pick up some rocks in Mars and see some signatures of life, you can get the ideas from here. So we worked on sites, and I say we because it was a team of Canadian and American biologists, part of a NASA team, and you might want to talk to Michelle later because she was telling me she's worked on things like this as well, and she'll be able to answer more questions than me. But there are some really cold springs in Axel Heiberg Island where there's permanently flowing water even in the winter, where microbes live and thrive. And I also worked on some glacial lakes, likewise, that are really cold and they're covered in really deep ice covers. And you've got microbes surviving because they're using mixotrophy. They're being very clever. They're finding amazing ways to survive that we just don't see back down here in temperate and tropical regions. But I've got a silly question here. Why do you think it's safer to work in the Antarctic than the Arctic? Anybody? Go ahead. Good, no bears. Yeah, you don't get polar bears running off with your field equipment, and this is a real photo. The polar bears did turn up, and they did steal our stuff, and they did break our gear, and we did carry guns in order to work safely in the Arctic, and you don't get that in Antarctica. So when I'd finished in Germany, and I went to become a lecturer at this at a university in Scotland, and I was based at the Scottish Association of Marine Science, I carried on Arctic research, but also Antarctic research. And that took me back to Antarctica again, to another space analogue. So you've seen this map before. You can see I was at Davis before. Now I'm going to bring you to McMurdo, which is the American station down here, and to the dry valleys, which is one of the most extraordinary places in Antarctica. It's another ice-free area, like the Vestfold Hills, but this time it's 4,800 square kilometers. It's much, much bigger, and it's the largest ice-free area in Antarctica. And we stayed in a station there that was run by McMurdo. It's a base camp called, these sort of structures are called polar havens, and you can have a look inside about where we worked. But it was a truly mind-blowing place. I'm a rock geek, and every rock just blew my mind because it was carved out by the winds. The dry valleys are pretty much the most extreme dry environment you can get. The average temperature is minus 30. It can get as low as minus 69.1. The coldest temperature ever recorded in Antarctica wasn't here. It was Vostok Station at minus 89.2, but minus 69 is brutal. I didn't experience there. People don't winter in the dry valleys for those reasons. It's so dry because of the way the winds are displaced by the Antarctic mountains that are nearby, that glaciers actually evaporate. They don't form, they evaporate because the moisture in the ice just disappears into the atmosphere. We come back, you can see there's a bit of an extreme theme coming on. This is a place called Blood Falls that's near one of those glaciers, the Taylor Glacier. And underneath the glacier, there's a very deep subglacial lake. there's no light down there. There's very little organic carbon. So the microbes there do something really, really cool. They take the very rich iron and sulfate from the water, and they do what's called chemoautotrophy. So they're using chemicals to create their own energy. And as a process from that, they convert the iron that they're collecting into another form of iron, so iron 2 to iron 3 again. And what the result is this really red chemical. So you can see the parallel with what I was doing in Germany, that red color because of the iron in the lakes. You've now got it being spat out of the bottom of a glacier, because when that lake is flowing and overflowing because of melt, it overflows these blood falls, as they're known, and you get this iron spilling out as a result of microbial activity. And this is the only place on the planet where you see this, visible. Usually it's all happening underground. But I actually didn't focus on blood falls there. What I did was work on another lake called Lake Bonny. It's a lake that's got a 4-meter layer of deep ice on it permanently. So it's a massive undertaking to do any work on this lake. You can see here there's a polar haven there, a little hut. And we have to do that because the problem in Antarctica is, an esky or a cold box, ice box, whatever you call them, where you come from. In Antarctica, you use those to keep samples warm. You do not use them to keep your beer cold. And it's the same with the polar haven. We can't do anything without somewhere to keep our water samples from freezing because we can't work outside. And you can also see we've got generators on the ice. That's because we can drill a hole in the ice, and it takes us a long time. It can take us a whole day to create a hole in the ice through that 4 meters of ice. But then we have to put a heating coil down to keep it unfrozen. So you can just see here with the generator with power going to this heating coil that's in the water. One of the augers from the ice core that's being used to keep the cable up out of the water. So you probably can't with the light see it that well, but trust me, there's a heating coil in there. But even so, every night, overnight, that ice froze again. And you can see here, even in the hole, there's a big ice on there. So before I could do my work, I had to chip out lots and lots of ice again and scoop it out of the way every day to take samples. And once again, I was working on really cool microbes here, microbes that were mixotrophic. This rig in the middle, this team I was working with, we had semi-permeable tubes, dialysis tubes these are, filled with water from the lakes that had cultured algae in them, and we'd lower that whole rig into the water and incubate it for many weeks, every day, pull it out, take a subsample, put it back in. And again, we were able to demonstrate we had a thriving community of organisms that were surviving because they were very versatile in the way they got their nutrition, the way they worked around the difficult environment they were in. But at this stage, I'm sure you're pretty sick of small things. These are diatoms, and this is one of the reasons that I was drawn to small things, even though I wanted to work on big rainforest trees, because some of the algae are covered in these silica frustules, and they're absolutely stunning under a microscope. And I remember the first time I saw them at university, I was like, wow, they're not boring, they're incredible. But what's also incredible is, I'm assuming that all of you, while I've been talking, have been breathing, haven't you? Yeah? Can't see anybody who's turned blue. There might be a few people who are asleep, but you're definitely not blue. Did you know that every second breath that you've taken while you've been listening to me has been created by these? So every second breath is created by the oxygen derived by phytoplankton in the oceans creating oxygen. So they're vitally important for us. They're also vitally important for these guys. Do you remember Bill and Will, the Antarctic krill from Happy Feet, or are you just too young for Happy Feet? Please tell me someone, yes, you do know Happy Feet. I was an adult when I saw Happy Feet, and I can't forget Happy Feet. It's just amazing. So Antarctic krill, they're what? We're talking about microbes. We said a 20 millionths of a meter long in size, billions of them. Then you've got billions of krill who depend on those. And krill, these are Antarctic krill, they get up to about 6 meters in size. What relies on the krill? Whales. And this is what I'm doing now, and what I have done for the last 11 years, is work on creatures like whales. This is the Antarctic blue whale, and they can grow up to 33 meters long, and they depend entirely on Antarctic krill. They can eat between 3 and 8 tons of krill a day to survive. So you can see how important those little things, hence the importance of my title, those tiny little things grow some of the biggest things on the planet. And my career has gone from studying really tiny things to really big things too. But it all ties together, so there is a link and a similarity between those bacteria and viruses which help grow the algae, which grow the krill, which feed the whales. And I'm going to tell you a lot more about whales and work tomorrow because we don't have time now. But I just want to finish with some extra tips that I think are really important. Mentoring. Now, I've stolen some clip art from various websites rather than creating my own terrible cartoon, but you might be lucky enough in your university setup when you do get to university, or even within school, to have a mentoring scheme. But regardless of that, find a mentor. They don't have to be the preeminent expert in the field. They don't have to be the best scientist on the planet, but they have to be someone you trust to give you good careers advice, to ask questions when you're thinking about taking a risk, to talk it through with. So find one. They're really important. And likewise, if someone asks you, could you be my mentor? Can I get some advice? Do help them out, because you pay it forward. It'll come back and help you in the end. And I'm not sure if you noticed that during my talk, it was really only early on that I was talking about I, probably when I was talking about my PhD. And later on, I talked about we and teams, because I think that's absolutely essential that you collaborate. Gone are the days where scientists were shut away in their ivory towers, just doing their own thing and following their own ideas. Now the biggest the global problems we've got to study and deal with, like climate change, require a whole planet of scientists to deal with them. You've got to work together. This is the team I work with at the moment. I'm program leader for a Southern Ocean Ecosystems program, which I'll tell you a bit about more tomorrow. I couldn't do anything without them. In fact, as a scientist now, as a program lead, all I do is push paper around. I get to do very little research of my own, but what I do is facilitate other people doing their great research. And we're a team doing it, because if I didn't push the paper around, they'd have to, and they wouldn't get to do their science. So you can see how it all comes around. And then you have to believe you can make a difference. Maybe there's one Nobel Prize winner sitting in this room, certainly not me. But even if you're not a Nobel Prize winner, even if you don't publish 1,000 scientific papers in your career, the tiny bit of research you do may make a difference. It might be that one puzzle that finishes the jigsaw. It might be the puzzle piece that goes in place that allows the next person coming on behind you to fill in the rest of the puzzle. You will make a difference if you work together and you stick to your guns and you follow your passions and use your skills wisely. And you will get there in the end. Never in my wildest dreams did I think I would be studying whales in Antarctica. I never ever thought, I think a lot of students would say, oh, I want to study whales, I want to study dolphins. I never ever thought that. I thought I might study rainforest trees. Now I'm in the Antarctic and I'm studying whales. And that's okay. I've achieved what I wanted to, because ultimately the science that I'm doing is with a magnificent animal in a fascinating place. And as you'll hear about more tomorrow, I'm making a difference, because where I work now at a government department, the Australian Antarctic Division is part of the Department of Climate Change, Energy, the Environment and Water. We have a direct line to government. The science we do directly informs the conservation management decisions they make about whales, about krill, about fish in the Southern Ocean. And so you can make a difference. And you will get there, even if now you think, well, I'm not quite doing what I want. It might just be a stepping stone along the way. So there's a bit of a summary of the tips. And depending on time, I've got something fun to show you. Have I got 5 minutes?
You're good. You've got about 20 minutes in total, including questions.
Okay. Oh, gosh. Right, if I keep talking, then you can't ask me any questions, and I'll be safe, won't I? Okay. So I've got my tips there. Don't be afraid to make chances. Your skills are transferable. Your skills and your passions can take you anywhere if you want them to, but likewise, you can stay in one place and still make a difference. That's absolutely fine too. I've got a massive travel log. I'm actually quite a homebody, but I've traveled all over with my science, and I feel very privileged, but I would have been just as happy staying at home. You have to walk away when you need to. One thing I didn't touch on with that one is, one thing you don't have to tolerate these days is any discrimination or bullying or poor behavior. Stand up to it, do something about it, call it out. But if you need to walk away from that sort of behavior, you do. Or you help a colleague do it. You do not put up with it. Find a mentor if you can. Collaborate. Exclamation mark on that one, because no science can be done without collaboration anymore. And that believing you can make a difference, and you will get to where you want to be in the end, even if you have to take a few side trips on the end. So for fun, and I'm quite happy to answer any questions about the science and the things I've talked about, but you may also want to just know what it's like in Antarctica and some of the silly things we get up to. But not so long ago, it was the winter solstice. Are any of you from the northern hemisphere? Have any of you traveled from? Okay, so it was the summer solstice for you, but we had the winter solstice. This is Mawson's expedition, well over 100 years ago, and they always had a tradition of studying, when they were in Antarctica, of celebrating Midwinter. That is a tradition that continues when you're down in Antarctica for a winter in Australian bases, but also all around the world. One of the stupid, crazy things we do, I'm going to show you a series of photos. So this is a real picture. This is how dark, this is this twilight. It's the lightest it gets in the middle of winter. For 6 to 8 weeks at Davis, the sun does not rise above the horizon. It's pitch black. This is on the sea ice, which is about 1.82 meters thick. So once it's got really solid, we use that as our highway. We can drive vehicles. In the summer, we've got helicopters that can drive us out to our field sites over the ocean. But once it's frozen, that ocean's frozen, we can get in a Hagland, we can get in a tractor, we can get in a ute, we can get on a skidoo or a quad, and we can zoom around to where we want to go. In this case, we've got a container on the sea ice outside the station. And then we get some pretty strong men. The women could do it too, but we didn't have any women tradies on station. It was just scientists when I was there. And they got some very big chainsaws. They like to be very competitive with their chainsaws. And first of all, they've drilled some holes. They've frozen some big chains in the ice. Then they've got these huge chainsaws and they've cut a big chunk of ice and levered it out. Now, this is a very overexposed photo because it wasn't as bright as that in reality. But they've lifted out this huge chunk of ice. Now, why would they be doing that? Anybody got any ideas? Sorry? Oh, maybe. So we're creating a nice hole in the ice. We've got a nice ladder there. Note that it's wood, not metal. There's a very good reason for it being wood, not metal. And then we scoop out all the ice that's constantly forming on the top. Literally, as we speak, it's -20, -25, and the ice is freezing. This is seawater. It freezes at -1.8. The air temperature is -20, -25. It's freezing instantly as you speak. And then we do things like this. Now, if the video works, it will probably let me down. Let's see. Please work.
Oh God, what am I doing? In focus! It's a mermaid.
Note that the person helping me out is in a full-on freezer suit with balaclavas and gloves, and I'm in my bathers. Now, this is a tradition. I've done it in the Arctic, I've done it in the Antarctic, and as have all my colleagues and teams that we've done it. It's really what you have to do, and it does happen in Hobart as well, but I'm not game to do it in the cold waters of Hobart. I'll only do it in Antarctic. And that's it. That's the sort of thing you get to do in Antarctica, and so I'm going to leave it there. And I have questions, and I will answer almost anything about what happens in Antarctica. Thank you very much.